welcome everyone at the second Yellow webinar organized by FIF in 2026. Today it's a great pleasure for us having with us Professor Neil Henderson. Professor Henderson is Chair of Tissue Repair and Regeneration at the Institute for Regeneration and Repair, Centre for Inflammation Research at the University of Edinburgh in Scotland, UK. Professor Henderson completed his medical education in Edinburgh and in London. He subsequently undertook a 3-year postdoctoral fellowship in hepatology at the University of California in the USA. And then he was awarded with a Wellcome Trust Senior Research Fellowship in Clinical Science that was focused on his current main field of interest, that will be also the topic of today's seminar, that is investigating the mechanism of organ fibrosis and regeneration using cutting-edge techniques as single-cell genomics. So it's now a great pleasure to give the floor to Professor Henderson for his seminar that is entitled Multimodal Decoding of Human Liver Regeneration and Repair. Please. Thank you.
Thanks very much, Christina, and thank you very much to you and the Foundation for asking me to speak today, and I'm looking forward to hopefully lots of questions at the end and just hearing more about everyone's research also. So my talk today is essentially broadly discussing some of the single-cell and spatial transcriptomic work we've done in the context of human liver regeneration and fibrosis over the last few years. And I'm going to start off with a story about liver regeneration, and then in the second half of my talk more about some of the spatial technologies that we've been trying to harness over the last couple of years. Okay. So in Edinburgh, we are the National Liver Transplant Unit for Scotland, and so we have lots of liver cases come through our hospital here. And so over the last 10 years, we've set up a single-cell genomics and spatial genomics pipeline. And broadly speaking, we look at both healthy human liver, or as healthy as we can get. We usually get that from colorectal metastasis resections, and also diseased liver, both chronic liver disease etiologies and the demographics of chronic liver disease, I'm sure, are very similar between Italy and Scotland. We see a lot of metabolic dysfunction-associated liver disease, also the biliary cholangiopathies, which you'll be very familiar with, PSC and PBC, also alcohol-induced, and this new classification of ALD as well. We study acute liver failure as well, and some of the data I'll show today was based on acetaminophen-induced acute liver failure and non-AT hepatitis. We've also looked at other rarer causes of acute liver failure. And really the top right of this slide is just showing some of the technologies that we use. So we do a lot of single cell and single nuclei RNA sequencing, and also spatial transcriptomic approaches, both unbiased, for example, Visium HD, and also biased pre-designed in situ gene panels using systems such as Xenium. All right, so front up, I wanted to acknowledge these 4 people who did an awful lot of work for this body of work that I'm just going to talk about for the next 5 to 10 minutes. Kylie's a senior wet lab postdoc in our group, and John, Jordan, and Andreas are bioinformaticians. So as many people will know with single cell work, it's very, very helpful if you can have some heavy bioinformatics support as well. So acute liver failure, many people on the call will know this syndrome well and will have looked after patients with acute liver failure. For those who haven't, it's quite a scary syndrome. It's one of the scariest, I think, in hepatology, because it can be unexpected and rapid onset and has a very high overall mortality rate of 30%. Acetaminophen, or as we call it more commonly in the UK, paracetamol poisoning, either accidental or non-accidental, is the commonest cause of acute liver failure in the UK and US. And also non-AT hepatitis is the next most common cause of acute liver failure in the UK. And really with non-AT hepatitis, we call it that because we don't know the cause, essentially. And as we all know, donor organ demand greatly outstrips supply for liver transplantation. And this has knock-on effects because if we need to transplant someone for acute liver failure, that means that someone on the chronic waiting list loses out on a liver and may die. So all of this has knock-on effects. So as you can imagine, our group and many other groups are interested in really trying to harness the inherent regenerative capacity of the liver. We all know how regenerative the liver is, but can we actually harness that even further to drive regeneration in settings like this and chronic liver disease? So what Kylie did initially was we wanted to understand more about the rates of hepatocyte proliferation in some of the common chronic liver diseases, and I've called it on the graph on the right NAFLD there. That's out of date now, it's MASLD. But these are explant cases for transplantation of alcohol-induced liver disease, NAFLD, MASLD, and PBC, comparing it to explant tissue from acetaminophen-induced acute liver failure and non-ATE-induced acute liver failure. And as you can see, despite this liver being severely injured and bad enough that we needed to transplant the patient, in the acute liver failure cases, there's actually quite a high rate of hepatocyte proliferation in the remnant hepatocytes that are still viable. So that liver is doing its best to keep its hepatocytes proliferating and regenerate. You can see in the chronic liver diseases, as you might expect, very low levels of hepatocyte proliferation. And clearly in acute liver failure, there's a very high amplitude injury there compared to, say, a grumbling MASLD injury at lower level. So just to remind people about acetaminophen-induced liver injury, you can see on the left here we have a healthy liver lobule, central vein in the middle, portal vein at the periphery. But with acetaminophen-induced injury, we see zone 3 necrosis, where the hepatocytes metabolize a lot of drugs and a lot of toxins. And in human cases, that necrotic area can expand right out to nearly the portal triads and wipe out a lot of the lobule. So previously, myself, our group, many others thought that hepatocyte proliferation was really the major driver of liver regeneration. And I'm going to talk a bit about how we've looked at that and how we think that is partially true, but not the whole story. And also, just to orientate you, the PNR and the RVR you see in the top right schematic there, PNR stands for perinecrotic region and RVR is remnant viable region, i.e. just the surviving hepatocytes. So Kylie initially looked at necrotic area in the acetaminophen-induced acute liver failure explant tissue. And you can see here that, as mentioned, the necrosis area can range from anything from 30% all the way up to essentially wiping out all the hepatocytes. So quite a range of hepatocyte necrosis. And it's interesting that these patients fulfill the King's College criteria for transplantation, but they exhibit on pathology, when you look at the tissue afterwards, quite a variable rate of hepatocyte necrosis. Kylie then looked at hepatocyte proliferation. And again, this is the remnant viable region in the middle, the surviving hepatocytes in the perinecrotic region. And what she found was that most of the hepatocyte proliferation was in the perinecrotic region, just around that area of dead tissue. So what we did was we reached out to various people that we collaborate with and work with. Acute liver failure is rare, and so we had a few frozen tissue cases in the freezer biobanked, but not that many. And so we reached out to collaborators in Cambridge and Birmingham in England and the transplant centres there, and also the US Acute Liver Failure Group, which for 20 years has collected acute liver failure tissue, blood, etc. And Will Lee in Dallas coordinates that. Sadly, the NIDDK pulled funding from that, I think maybe 18 months ago, but that's been a fantastic resource for studying acute liver failure. So what we then did, and sorry, just to mention, so single cell RNA-seq, came along nearly 10 years ago now, and that usually involves, not always, flow sorting of fresh cells from cases. And also this syndrome is so rare the beauty of single nuc-seq here was the fact that we could take frozen samples that might have been in -80s around the world for a good 5, 6, 7 years, get them over to Edinburgh and perform single nuclei RNA-seq. So it opens up frozen biobanks in essence. So what we did was we generated a pan-lineage atlas of human liver regeneration with single nuclei RNA sequencing. You're essentially spinning down a hard centrifuge of all the nuclei. And so what this allows you to do is get a real broad look at the different cell types and really a pan-lineage atlas. So we sequenced around 70,000 nuclei for this project, both healthy human liver and also acetaminophen and non-ATE induced acute liver failure tissue. And what we then did was we took a deep dive on the different lineages, but one of the lineages that particularly caught our eye was hepatocytes. And you can see here that when you subcluster the hepatocytes, there's cyclin hepatocytes to the top right on the UMAP, but there's also various different subpopulations of hepatocyte. And our eye was caught by the green subpopulation at the top, which I'm naming ANXA2 positive because that was one of the differentially expressed genes that marked that population. And when you look at the graph on the right showing relative contribution of the different hepatocyte subpopulations, you can see that the ANXA2 positive population was very much expanded in acute liver failure compared to healthy human liver. And when we did a few different flavors of GO analysis to try and understand, try to pick apart initially what type of function these ANXA2 hepatocytes might have, it was very strongly suggestive of a migratory phenotype, which, normally we don't associate hepatocytes with much movement at all. And so this piqued our interest and we started to look into this further. So maybe a couple of years ago now, maybe 3 years ago, the limit of technology was around 100 plex SM fish. This is obviously, that number's gone up a lot in the last couple of years. But at that point, we generated a migratory hepatocyte signature. So I've not proved to you that these are migratory, but the GO terms were smelling of a migratory phenotype. And so we took the gene signature from the single nuclei-seq data for that subpopulation and generated a high-plex SM fish gene list that we then did in situ on. And what you can see here is that these hepatocytes marked in pink sit at the edge of the necrotic area. And so we started to get more and more interested in these hepatocytes that were sitting just at the edge of the necrotic area. And of course, with this gene panel, you can compare it to healthy tissue and also mark the other major lineages. This is protein staining, and ANXA2 I mentioned was a differential marker. ANXA2 is also expressed on other cell types in the liver, such as different types of mesenchyme or leukocytes. So what Kylie had to do here is do a co-stain with HNF4 alpha to identify hepatocytes. And what she found was that ANXA2 was a membrane stain in hepatocytes and was upregulated in acute liver failure compared to healthy, as she's quantitated on the right. And again, at protein level, these hepatocytes mainly resided at the edge of the necrotic area. Zooming in with the multiplex immunofluorescence, what we could see was that these hepatocytes had ruffled membranes with extended lamellipodiae. You can see on the left, and also when we do F-actin staining, this again showed this ruffled membrane to the edge of these hepatocytes. So they looked very much different from their normal neighbours, as it were. And because the transplant unit in Edinburgh has been open over 30 years now, we have quite a lot of acute liver failure cases in the FFPE biobank. And so we looked with ANXA2 staining and HNF4 alpha staining in multiple etiologies of acute liver failure. I've listed here hepatitis A induced acute liver failure, Hep B, terfenadine as an idiosyncratic drug reaction, and we've transplanted 2 or 3 patients for ecstasy-induced acute liver failure over the years. And in essence, what we found was we could see this hepatocyte phenotype in acute liver failure cases irrespective of etiology. So I've talked there about the human acute liver failure setting. Now, with human, in essence, we have healthy tissue, and then we have the livers that are so damaged that we need to transplant the patient. How do you start to pick apart the mechanism of when this hepatocyte population is appearing, when it might be disappearing? And with the human data, one of the things we did was we collaborated with Rajiv Jalan down at UCL in London, who has transjugular biopsies of acute severe liver injury, and that was very helpful to look at the ANGSA2 positive population in those cases as well. But to really get at the temporality of this process, we moved to mouse because that allows us to pick apart the temporal ANGSA2 expression and also look at rates of proliferation relative to ANGSA2 expression. So that's what I'm going to talk about for the next couple of minutes. The mouse model of acetaminophen-induced acute liver injury, you know, we all know the limitations of mouse models. We all do our best with mouse models to try and model human disease. I think this is one of the better models. It recapitulates a lot of the histological features of acetaminophen-induced acute liver injury. Of course, you can't make it as severe as, say, some of the human cases with full-on liver failure, but it is a very helpful model to try and understand more about acute liver injury and regeneration. So what we did here was, this is wild type black 6 mice, given acetaminophen 300 milligrams per kilo at time 0. We give that IP, and then Kylie did a heroic time course after that, harvesting tissue and cohorts of mice at these time points, and then we generated a single nuclei-seq atlas, just like we had done with the human tissue. And zooming in top right on the UMAP for hepatocytes, you can see this green tail that was ANGSA2 positive. And if I can draw your eye to the bottom right, the beauty of developing a time course obviously is that you can look at progression, and so we can generate things like this GIF video, which is just running through time points, looking for when ANGSA2 gene comes on in these hepatocytes and when it switches off. And then when we did GO analysis, bottom left, again, similar to the human ANGSA2 positive hepatocytes, we saw lots of terms that were enriched for cell movement, actin filament organization, etc. So this is immunofluorescence zooming in on these hepatocytes in the mouse model. Again, you see ruffled membrane and lamellipodiae. And when Kylie quantitated these ANGSA2 positive hepatocytes, they were very much enriched in the perinecrotic region. We saw less of them in the remnant viable region compared to the human condition, and there's obvious reasons for that in terms of, we cannot model exactly the human condition. But what she saw was that these hepatocytes peaked around 36 to 42 hours after acetaminophen injury in mice. And this was very helpful to us because we wanted to understand when we might start to really see these hepatocytes in the mouse model and zoom in on them. So this is just some lineage tracing. As many people on this webinar just now will know, there's been lots of discussion over the last 10, 15 years about origin of hepatocytes, and clearly it can be mouse model dependent, human disease condition dependent, but we wanted to ask the question of where do these ANGSA2 positive hepatocytes come from? So we used this viral tracing system, adeno-associated virus 8, with thyroid binding globulin to drive CRE, so it's very hepatocyte specific. It's also very efficient, so you can get 99% or greater recombination of hepatocytes to turn them red, RFP with a reporter here. So we gave the virus to induce red hepatocytes and then gave acetaminophen and tracked that. And in essence, what we found with this model, this acetaminophen-induced injury model, was that 100% of new hepatocytes were coming from adult hepatocytes. And then Kylie very kindly quantitated this in the context of ANGSA2 positive hepatocytes. And as you might expect, given the graph I've just shown you on the previous slide, and it was very good of Kylie to count this nonetheless, we found that basically all ANGSA2 positive hepatocytes were coming from adult hepatocytes, so not a stem cell source by the look of things with this data. We're confident that these new hepatocytes were all coming from adult hepatocytes. So this slide, I'm just going to pause on for a minute because I think some of the data here's hopefully interesting for you. So we wanted to look at the temporal dynamics of wound closure around that zone 3 area over time in the mouse model. So in red on the graph on the right, you can see percentage necrotic area. In this mouse model at 300 mg per kilo in a black 6 mouse, necrosis peaks around 20-25%. You can see that at 30 hours, and then you start to get wound closure. And what struck us was when Kylie quantitated hepatocyte proliferation was that over 50% of wound closure had occurred before hepatocytes even started to proliferate. So there were mechanisms at play here that were not hepatocyte proliferation simply driving wound closure. And this piqued our interest further. Another way of looking at this is in collaboration with Tom Bird, who works at the Beatson in Glasgow and also Edinburgh. We set up this experimental design. So again, black 6 mice, gave acetaminophen at time 0, and then these mice had BRDU in the drinking water. So you can trace essentially any hepatocyte that's gone through cycle because the mice are drinking BRDU in the drinking water. And just to draw your eye to the uninjured micrograph at the top, glutamate synthetase, a really nice marker for the first 1 or 2 rows of hepatocytes around the central vein. And as you know, when you give acetaminophen, you wipe out the zone 3 area of hepatocytes. And so what that allows you to do at 14 days, when the liver has completely regenerated, is count the number of cells around the central vein, the new hepatocytes, that have, or should I say, I'll just simply say the hepatocytes around the central vein, count how many of them have incorporated BRDU or not. And the kind of key take home from this experiment was that around 3/4, even higher in female mice, of the glutamate synthetase positive hepatocytes at day 14 were BRDU negative, suggesting that those hepatocytes had got back to the edge of the central vein by means other than simply proliferating and dividing and filling in the pothole, if you like. So again, suggesting that the majority of hepatocytes in that area didn't derive from hepatocyte proliferation. So there was lots of smoking guns and data pointing at something going on other than hepatocyte proliferation in terms of wound healing with these livers. So what we then did was we collaborated with Leo Carlin and his group. And Leo is also based at the Beatson, and Leo is expert in intravital imaging, mainly of the lung, but we convinced him to come south of the diaphragm down to the liver, and he very kindly was happy to do that. And so we're going back here again to the AAV8 TBG Cree mouse with an RFP reporter mouse, so you can turn all hepatocytes red. And then I'll mention again the data that Kylie found and quantitated, where the ANGSA2 positive hepatocytes were peaking around between 36 and 42 hours. And that was super important for us with this intravital imaging experiment, because that then allows you to focus when you image. And intravital imaging can generate a lot of beautiful pictures, but if you don't know when to look, that can make your life a lot more challenging. We can only image for 6 hours under the Home Office licenses in the UK, which I think is absolutely perfectly reasonable, because the mice are under anaesthetic for a long time. So we're limited to 6 hours. And so we filmed between 36 and 42 hours via this system. So this is 24 hours after acetaminophen, where we did some initial pilot studies just to really look at necrosis in these livers. And you can see here it's almost like a sinkhole where you get hepatocyte death in zone 3 around the central vein. And obviously it's in 3D, so you just get this widespread death of hepatocytes. And then this is between 36 and 42 hours. And so you can start to see, hopefully you can see the wound edges beginning to fill and close. You can also see some hepatocytes at the edge of the viable epithelial sheets, extending lamellipodiae with ruffled membranes and moving. And when we zoom in on that, you can again start to see this closure of wounds. And because we were so used to looking at liver sections in 2D, we had to kind of get our head into thinking about, you'll see some hepatocytes coming up from below. And obviously, when one thinks about it, it's a 3D injury, so it's going to be a 3D repair. And so you can see the hepatocytes beginning to fill in these holes. This is just a further close-up on a hepatocyte. When we looked at these migratory hepatocytes, they were never lone wolves, as it were. They were never on their own. They were always at the edge of the epithelial sheet, very akin to epithelial healing in the skin with keratinocytes. And the other thing you can see is much rotation of nuclei. And I was kind of unaware of this until I had a read about it, but because of the actin fibers are tethered around the nucleus as well, what happens when cells become migratory is that the nuclei start to rotate. And apologies for cell migration aficionados in the audience, but that was a new one on me. Okay, so we then also used, as part of the intravital imaging studies, a mouse called an MTMG mouse, which is a dual reporter. So simply, these mice have, any of the cells have tomato fluorophore membranes. Then when you add Cre, it'll switch that cell that responds to Cre under a given promoter to GFP. And so we again use this hepatocyte Cre AAV8 TBG. And the reason we use this MTMG mouse is because it gives you very distinct membrane-only fluorescence. It's incredibly useful for cell segmentation and segmenting all the hepatocytes. And so Kylie, along with a guy called Fred Vercoque, who is French and expert in the informatics analysis of intravital imaging, which, as you can imagine, is non-trivial. Fred worked with Leo before he went back to France to start his own group. So Fred and Kylie did a lot of work on the intravital analysis of these mice. And so what you can do is you can analyze thousands of hepatocytes in one mouse, and each dot here is a different mouse, but in essence you build up data sets of thousands of hepatocytes. And so this is comparing degrees of hepatocyte mobility in both the perinecrotic region and the remnant viable region. And in essence, you can see lots of different flavors of readout here, but the short summary is that mobility of hepatocytes was greater in the hepatocytes just at the edge of the necrosis compared to, if you like, in the back rows of the more normal hepatocytes distal to the injury site. You can also carefully measure cell shape. And if you imagine prolate and oblate measurements are like squeezing a basketball that way or that way, and you can use various analysis approaches to get at that. And what Kylie and Fred found was that the hepatocytes at the edge of the necrotic region showed more changes in shape in lots of different parameters compared to the remnant viable region. Another question we were interested in was, could this wound closure, if we didn't think it was largely driven by proliferation, could it be driven by hepatocyte hypertrophy? Could it be that the hepatocytes are literally getting bigger and squeezing each other and squeezing that wound closed just by hypertrophy? And so again, Fred and Kylie looked very carefully at hepatocyte volume over thousands of hepatocytes in different mice. And the short summary of that data was that the wound closure was not secondary to shifts in hepatocyte size in the perinecrotic region compared to the remnant viable region. So I've mentioned ANGSA2, which full name is annexin-A2, and that was a really helpful marker of this hepatocyte subpopulation. And as we all know, genes can be helpful markers. It certainly doesn't mean they're functionally important. But when we started to dig into what this gene was and what it did and what it had been studied in settings other than regeneration, it sounded quite interesting. So annexins, large family of calcium-dependent phospholipid binding proteins, and I'm not going to list all the diverse cellular processes that it's been linked to, other than to say there was clearly a lot of work had been done on cell motility with this molecule, particularly in colon cancer. And so that again got us thinking, could this be functionally important in liver regeneration? So to interrogate that, I'm not going to go through the in vitro data we did. We knocked down ANGSA2 in hepatocyte cell lines and primary hepatocytes, but this is an in vivo experiment we did towards the end of that body of work. So again, acetaminophen-induced injury, and we used an AAV8 short RNA virus to knock down ANGSA2 specifically in hepatocytes. And what Kylie found was that when she knocked hepatocyte ANGSA2 down, wound closure was abrogated compared to scrambled short RNA control following acetaminophen injury. So in terms of, just to summarize that body of work, what we think might be happening is the following, and I'm at pains to say we've done a lot of the mechanistic work in mouse, and one is comparing this to human, but what we think might be happening is that when you develop necrosis in the acetaminophen injury model, the liver is actually prioritizing wound closure and restitution of normal microarchitecture over and above hepatocyte proliferation. So what we think is happening is that epithelial sheet is trying to close the gut-liver barrier as soon as possible. And as we all know, you all the time have pathogen LPS coming in from the gut. We know that in acute liver failure, the gut is leakier than normal, so it will see even more bacteria in the liver, more LPS. And so we think that the liver is very much trying to close that wound quickly and regenerate a barrier, as it were. And then afterwards, hepatocyte proliferation can kick in and start to regenerate the lost liver mass in terms of hepatocytes. Now, clearly, the kinetics of this will vary in terms of the amount of initial injury, more or less severe. But we think that this is perhaps what the liver is trying to do, because we know that in acute liver failure patients secondary to acetaminophen injury in humans, if you can't transplant those patients, about 90% of patients will die of sepsis and multi-organ failure. So we think this barrier is incredibly important in terms of the long-term outcome of the patient. Okay, just to switch gears a bit, so I'm moving away from acute liver failure to liver fibrosis here, and I'm going to be talking a lot about the spatial transcriptomics approaches we've used over the last 2 or 3 years to try and understand more about how the human liver scars and regenerates. So we published this paper 7 years ago now, time goes by. This was single cell prior to any spatial transcriptomics. And so what we've been trying to do is basically layer on, as well as single cell and single nuke, the spatial component. And I'll talk a bit about how we've tried to integrate those different types of technologies and approaches. And sorry, the other thing I wanted to mention with this is, this was highly laborious for the guys in the lab in terms of you postulate a new subpopulation, say scar-associated macrophages, but you then have to validate at protein level with immunofluorescence staining with lots of different antibodies what you're seeing in the gene level data with the UMAPs. So hopefully spatial transcriptomics will take away a lot of that slower work where you have to, rightly so, but laboriously go through different antibodies, testing them all. So many people will be well aware of this step change in the single cell genomics landscape, but in essence, what we're about with spatial transcriptomics is trying to use these technologies to understand two things that are different from single cell. So single cell, we have to dissociate tissue, collagenase digest the tissue, FACS sort it, etc. You've lost all spatial context, and also those cells may well have changed to some degree because of the isolation process, whereas spatial transcriptomics, we should be able to measure gene expression in situ where the tissue's not been anywhere near as disrupted and the cells are still in close proximity to their usual friends and neighbours in the tissue. And just down the bottom there, we published a review on this last year if people are interested in learning or reading a bit more about spatial genomics. And this was a kind of pan-organ review, not just liver. So the different spatial platforms we've been using are twofold. So the one on the left is Visium HD, which is sequencing based, and this is unbiased. So we're just trying to measure all genes possible. And the one on the right is called imaging based broadly, but it's in essence very high plex in situ RNA-scope, and we use the Xenium system. And so what we've been doing over the last 18 months or so is really getting to grips with the informatics analysis of these types of spatial approaches. This is just an example of two algorithms that we've been using, RCTE and bin to cell, for Visium HD analysis, and we've found that these two combined approaches, with having a base launch pad of single cell or single nuclei seq data, is very, very helpful. And I can't stress enough, if you have a really nice high quality single nuke or single cell data set to do your spatial analysis from, that is hugely helpful. And by that, I mean you've carefully annotated the different subpopulations in the single cell level data. That is really helpful for spatial interrogation. This is just a maseled explant liver section here, and you guys know very well, nodular hepatocytes in the middle with scarce septae around the edge. And what you can do is start to drill down into the epithelial subpopulations and also the mesenchymal subpopulations. And so what our approach, and everybody does it differently, I'm sure, but our approach has been to take unbiased approaches such as single nuke-seq, and that we have a large MASL single nuke-seq data set, and combine that with technologies like Curio Trekker, which is another barcode approach and is unbiased, and also Visium HD, to then inform custom panel design for your more focused spatial approach with imaging-based in situ spatial profiling such as Xenium. And I'm giving a big shout out to the informatics guys here, because spatial analysis is a lot of work and a lot of iteration. And I think in our group we're beginning to get there with how best to harness the spatial data, but it's informatics heavy. And of course the interfaces are getting better and better for interrogating spatial data, so that a wet lab, for example, PhD student can start to really analyze it via the 10x based interfaces. But of course, you often want to take it a little bit further. So having high quality informatics support really helps you get the most out of the data. So you have literally millions of cells in these sections. And so this is just to give you an example. So here we're using Xenium to look from uninjured liver, human liver, through the F grades all the way to cirrhotic explant. And what that gives you is an awful lot of cells, and therefore an awful lot of niches and neighborhoods, even within one section to interrogate. So I think there's two things here. Each of these sections can cost €5,000. So it's very expensive. But I think in a good way, we need to remind ourselves, or this is my own personal feeling, you don't need to do that high end because you have so much information in each of these sections. So there's a real balance here. I mean, in a good way for all of us, there is a limit financially on what we can spend on these technologies. But I really don't think we need to do crazy high end at all to get the most out of this type of data. So this is a healthy human liver, and what we can start to do is just really annotate each of the different populations. And so obviously one of your first questions with this type of data is, does it make biological sense? Is it largely in agreement with what we know about liver biology? And we always sense check these types of technologies when we start using them to ensure that. And you can start to subdivide the hepatocytes into portal and central. You can start to look at different subpopulations of endothelia or mesenchymal cells or macrophages. This is an F3 MASL liver, so obviously quite a bit of scarring at the F3 level. And again, you can start to pick out scar-associated macrophages or endothelia and start to drill down on your particular subpopulation of interest. This is decompensated cirrhosis. So this is a patient who's been transplanted for MASLed. And again, we start to really get into the vascularization changes that occur with the scar-associated endothelia and all the various different leukocyte subpopulations that you see in decompensated versus, say, an early F1 MASLed liver. This is mesenchyme, where again, we're getting into subpopulations, and even beyond this, I'm not showing it here, but we can get beyond, say, for example, stellate cells or fibroblasts and start looking at the subpopulations and where they sit within the liver in their various niches. This is just proportionality. So with this type of data, you can start to ask the question of, how many hepatocytes as a proportion are there in an F0 liver versus an F2 liver, and so on through to explant. And again, the beauty here is that once you've annotated all the major lineages and their subpopulations, is you can start to ask questions of this data from various angles. If you're into NK cells or if you're into Kupffer cells, you can really salami up this type of data to try and answer your specific research question. And broadly speaking, what can spatial tell us? So I think, there's a long list here, and these are just some of the main headings that we included in this review. But clearly what this allows you to do is to look at, as mentioned before, cells in situ, therefore neighborhoods that actually are reality and not an artifact of, trying to to backfit things once you've done single cell RNA sequencing. So it's very good for looking at neighborhoods. I think it allows you to look at what to the human eye looks like morphologically, histologically normal tissue, and look at that at gene level. And of course, perhaps unsurprisingly, areas that look normal, even in a very fibrotic liver, of course, are not normal. So you can use it to spatially look at transition towards a very fibrotic region of the liver, for example, and what changes as that happens. And you can also use it to interrogate ligand receptor interactions in situ between your two favorite cell types. You can really use this data to try and interrogate what the key signaling mechanisms might be between cells in close apposition, and the list goes on. So how do we try, and this slide's just about, trying to ask ourselves the question, how do we translate these types of technologies? It's very easy to say, single cell, spatial, they're shiny baubles, surely they'll be useful in medicine, full stop. But of course that's not the case. And, the whole community is thinking about how we try and translate these technologies. So I think for sure, therapeutic target identification for me is a reasonably straightforward target identification. I think is a real use of this. The other thing that we should aim to do is use this type of technology to stratify disease more accurately, which would help our clinical trials. We can also use them to understand mechanisms of action of drugs. Some of the drugs in phase 2 in Masold, the people who have these drugs are very candid and say they're unsure of the mechanism of action to a large extent of some of the drugs which have been efficacious in phase 2, but we don't really understand why. So we can use this type of technology to help understand that. The other thing I'd mention is I think we need to get, and it's not even think, it's simply we need to get better than liver biopsy for staging liver fibrosis and understanding liver fibrosis. So can we use single cell type technologies to derive precision markers to understand more about the biology, and how do we do that? So molecular imaging is definitely gaining in popularity. For example, PET tracing now, I can certainly speak for the NHS, the healthcare system in the UK. We're not going to be rolling out PET tracing in liver fibrosis patients anytime soon. However, I do think we will be rolling out PET tracing in this context in the coming years in the context of clinical trials. So I think this whole area of using molecular imaging in clinical trials to get earlier signals of efficacy will be both popular with the biopharmaceutical industry and also us as investigators trying to get readouts on whether a drug's going to work much earlier than running long, expensive trials. The other advantage there is that you can get a whole organ readout with these types of technologies as opposed to a biopsy, which is just 1/50,000th of a liver. So I would like to just acknowledge many people, Kylie, John, Jordan, and Andreas for the work in the Liver Regeneration Project, many people in my group for a lot of the spatial data that I was showing there, the patients for giving us bits of their liver. We couldn't do any of this without them, and also my surgical colleagues and the transplant coordinators for all their help. Got a list of collaborators who were fantastically helpful for this work. I'd like to thank all the funders, and also we have QR codes down the bottom. Whenever we publish our data, we make it both publicly available but also browsable in these atlases where you can search for your favorite cells or genes in the various different atlases. And just to say thank you very much for your attention. I'd be delighted to take any questions. Thank you.